All right, let's dive straight into this explainer and see exactly how artificial intelligence managed to solve one of biology's most notorious mysteries under the hood. For decades, the protein folding problem was this massive computational wall that literally no one could climb. But then came AlphaFold. Today, we're not just looking at the end results. We're going to pop the hood, open up the engine, and see exactly how Google DeepMind's system actually works. Here's our roadmap for this explainer. 1. The protein folding problem. 2. Reading evolutionary clues. 3. The Evoformer engine. 4. Building 3D structures. 5. The final physical polish. And 6. A new era of biology. Section 1. The protein folding problem, a 50-year grand challenge. So for 50 years, figuring out how proteins fold was this monumental grand challenge. I mean, proteins underpin literally every biological process in every living thing, right? But mapping just one of their unique, highly complex structures in a lab using methods like X-ray crystallography could take years of painstaking research. We're talking hundreds of thousands of dollars just for a single protein. Because the core problem is basically this: how on earth do we mathematically translate a flat 1D sequence of amino acids into the complex 3D shape that dictates exactly what that protein does? The number of possible shapes a single protein could theoretically take is so astronomically huge that calculating them all one by one would literally take longer than the lifespan of the universe, which is crazy. But that's the exact barrier AlphaFold had to break. Section 2. Reading evolutionary clues. The data input. Okay, so AlphaFold starts by taking your target protein sequence and scouring massive databases like UniProt to find similar sequences in other species. We're talking everything from humans to horses to bacteria. Using software tools to build what's known as a multiple sequence alignment, or MSA, it creates this 2D table. It essentially lines up all these related sequences so the AI can play detective and spot evolutionary patterns. And here is the crucial takeaway. If we look down the columns of that MSA table and see that when an amino acid mutates in one spot, another amino acid in a completely different spot always mutates with it across millions of years of evolution, well, what does that tell us? It's a massive clue. It tells the AI that these two seemingly distant amino acids must actually physically interact in 3D space. They're functionally linked. Section 3. The Evoformer engine, the core logic. Now we get to the heart of it. Inside the 48 identical blocks of the Evoformer, the system uses a brilliant dual representation setup. On one side, you've got the multiple sequence alignment tracking evolutionary history. On the other side, you have a pair representation, which is essentially a 2D map tracking the physical distances between every single pair of amino acids. The real magic of the Evoformer is that it's constantly trading information back and forth between these two, updating each representation based on whatever it learns from the other. Inside this continuous loop, the data passes through some major processing stations. It uses attention mechanisms to refine the evolutionary data, passes that new intel to the distance map, and then applies strict geometric rules to make absolutely sure those distances actually make sense. To process all that evolutionary data without completely melting the computer's memory, AlphaFold uses something called axial attention. Think of it like a highly efficient, kind of fuzzy database search. Instead of trying to look at every single piece of data all at once, it first searches horizontally, finding relationships between amino acids within the exact same sequence. Then, it searches vertically, looking down the columns to see how a specific amino acid has evolved across different species over time. Once that evolutionary data updates the physical distance map, AlphaFold applies triangle inequalities. This mathematical step is absolutely vital. Think about it. If the AI guesses that amino acid A is right next to B and B is right next to C, then A and C simply cannot be a mile apart. By running complex triangular self-attention algorithms, the system enforces the physical reality of actual space onto our distance map, strictly preventing any impossible knotted-up geometries. Section 4. Building 3D structures, the assembly line. So here, the system relies on something called invariant point attention, or IPA. AlphaFold represents every single amino acid in the protein backbone as a rigid 3D triangle. Initially, they're all just clumped together at the origin point, which the creators jokingly call the black hole initialization. But then the IPA kicks in. It acts like an intelligent robotic arm, treating each rigid 3D triangle as an individual body and shifting and rotating them in local 3D space. Now, the true power of IPA is that it doesn't just guess randomly. Not at all. It seamlessly combines the sequence embedding from our evolutionary data, those pair representation distances we just figured out, and the current state of the 3D structure. It merges all these clues together to continuously snap the protein backbone into its proper 3D configuration. And it actually recycles the structure through the network multiple times to really perfect it. Section 5. The final physical polish, OpenMM. To ensure the protein strictly obeys the actual laws of chemistry, AlphaFold feeds this AI-generated structure into a physics simulator called OpenMM, utilizing the Amber force field. This step physically relaxes the structure. It's essentially an energy minimization process, just slightly shifting atoms to fix any clashing boundaries, correcting unlikely bond lengths, and resolving any stereochemical violations, which, by the way, is just a fancy way of saying it ensures two atoms aren't physically overlapping. It is the final coat of paint that makes the model scientifically sound. Section 6. A new era of biology, the impact. What took us months and years to do, AlphaFold was able to do in a weekend. This quote from Professor John McGeehan really hits the nail on the head. It highlights the human awe behind all this math. Tasks that used to dictate the entire duration of a PhD student's thesis were suddenly being spit out by AlphaFold before Monday morning. By open sourcing these predictions, DeepMind effectively saves scientists hundreds of millions of years in cumulative research time. And we really need to appreciate the sheer scale of this. Before AlphaFold, 60 years of experimental methods had mapped roughly 17,000 protein structures. By applying this AI pipeline, the AlphaFold protein structure database has now mapped over 200 million structures. To put that into perspective, that is nearly every single cataloged protein known to science, across plants, animals, bacteria, and it's all available for free. The speed of this historic breakthrough is honestly staggering. In 2018, AlphaFold 1 shocks the world by placing first in a global structural prediction competition. Just 2 years later, AlphaFold 2 achieved atomic accuracy, officially solving the 50-year problem. And by 2024, creators Demis Hassabis and John Jumper, alongside David Baker, were awarded the Nobel Prize in Chemistry. And guess what? The factory is already upgrading. AlphaFold 3 has recently launched. It swaps out the Evoformer for a new, simpler paraformer architecture and actually incorporates a diffusion model. Because of this, it's not just limited to single proteins anymore. It can now predict how proteins fold and interact with DNA, RNA, ligands, ions, essentially mapping the interactions of all of life's fundamental molecules. So, we've seen how tracking evolution, passing it through dual representations, enforcing geometric constraints, and applying physical laws completely solved a 50-year mystery. But it leaves us with a pretty fascinating thought. Now that we have the complete 3D blueprints of life sitting right there on our hard drives, what incredible cures, sustainable materials, and biological discoveries are we going to build next?